patients with metastatic melanoma or kidney cancer told by their physicians that they were a few months from death. They would receive high-dose IL-2, and for about 4 or 5%, metastatic lesions would melt and they would be cured. I mean, it was just extraordinary. AlphaFold, it'll give you 50 low-confidence predictions. That obviously doesn't help you discover drugs, so we have no training data. Here's the clever bit. Go back to AlphaFold, or some version thereof. You've given me 50 low confidence predictions for what the structure of this protein might be. I now have data. One is a prediction, the other is data. And you end up with what looks like something that fits perfectly. So now you've got your teeth into something that looks real.
What's up, Andrew? How are you?
How's it going? Good to see you.
Good to see you.
Yeah, I've lived here for a long time now. I moved here in 2004.
Okay.
So over 20 years, yeah. But yeah, I hail from England originally.
What prompted the move? It wouldn't be company building or anything like that, would it be?
Yeah, it's all about biotech. Yeah, back in the day, you know, I was a academic working in the National Health Service. I was an academic nephrologist working in kidney disease, mostly inflammatory kidney disease, so renal transplant rejection and glomerulonephritis. Back then, and this is kind of hard to believe now, but nobody did anything in kidney medicine, right? There were no interesting drugs. My old boss in a highly academic institution, he was a great guy, he finally got his New England Journal paper, and the topic was, for systemic vasculitis, should I treat with Cytoxan for 3 months or 6 months, right? Which is a very important clinical question, but I think we can agree not scientifically bleeding edge. But that's kind of where the field was. So as I got into science and biotech, it became clear that oncology was where the action was. And so basically, since I moved into biotech in '99 and came to the US, I've been pretty much in oncology for most of that time.
Yeah, you were among the very, very first to work on cancer vaccines. That's...
Yep. It's been a great journey, obviously. So the field has
nice
been
and easy
evolving.
space.
Yeah, for sure, right? So we had some hubris back in 2015. So when I first moved to the Bay Area, I was working at Chiron, right? One of the original pioneers, right? They used to talk about the ABC of biotech: Amgen, Biogen, Chiron. Genentech was in there too, right? But one of the early players. And Chiron had bought high-dose interleukin-2. So that was an asset that belonged to the company. It was approved by some of the sort of pioneering oncology drug developers back in the day, who got this drug approved for melanoma and kidney cancer. There's a woman called Gwen Fife, who was famously involved in that. She went to Genentech and was very successful at Genentech. She's the one that actually was at Chiron getting that drug developed. Gwen, if you're out there, is still consulting for investment firms now. So high-dose IL-2 obviously activates the immune system, and I arrived at that company all bright-eyed and bushy-tailed in 2004, I think it was. And it was clear that the drug had a market, right? It was doing about 100 million, something like that, in the US, with a little bit in Europe. And it was miraculous, right? Steve Rosenberg had pioneered this thing. And it was truly a Lazarus-like drug, where you'd have patients with metastatic melanoma or kidney cancer who were told by their physicians that they were a few months from death. And they would receive high-dose IL-2, and for about 4 or 5%, they would mount these phenomenal immune responses, and metastatic lesions would melt and they would be cured. I mean, it was just extraordinary.
That is pretty extraordinary.
And there's a magic to that. I think that people don't fully appreciate that once you've seen that, particularly as a physician, you just don't let go of that very lightly. And so Steve obviously recruited this generation of investigators, both surgeons and physicians, because he's the head of the surgery branch at NCI. And so a lot of his followers were surgeons originally. And that's probably relevant because for things like TIL, you actually have to have surgical specimens that you pull T cells out of. So that's probably not unrelated. Anyway, he had this huge following. And so there were people who'd seen this, and they just became devotees of the power of immunotherapy. But it was clear that for 20 years, we didn't really know how these drugs worked. And that was clear to me at Chiron, that we just didn't have a very strong intellectual framework. How do you build on that effect, that amazing and miraculous effect, but it's only found in 5% of patients? We just didn't know how the drug was really working. Obviously, it bound to a receptor on T cells, and they sometimes kill tumor cells. That's kind of all we knew. So it was very hard to kind of put a scientific framework around that and think about life cycle management, all the usual things. The company actually got acquired by Novartis, and so I just turned away from it, to be honest, because it was just, I couldn't see an obvious path forward. right? How did you move forward with immunotherapy back then? There were vaccine companies around, but they basically were consistently failing. And you couldn't see a clear, logical path to something other than failure, right? Hope might get you there, but you know, that's not a very reliable strategy, as they say. And so I actually turned away into a small molecule lab and left vaccines for a decade. And nothing much happened, except for the folks, the devotees who didn't turn apostate like me. They stayed with it. And obviously Jim Allison is famous for this, as developing the next wave of immunotherapies, which was not vaccines, it was checkpoint inhibitors. Obviously in Jim's case, it was ipilimumab, and then out of Japan, obviously came the PD-1 antibodies. And those started to demonstrate that they were having similar miraculous responses in larger fractions of patients, particularly melanoma, of course, where the response rate for monotherapy was more like 25-30%, not 4 or 5%, and they were much better tolerated. So clearly a big step forward. And then famously they were put together by Bristol in the combination of Ipi-Nivo, and now you're at a sort of 45 to 50% response rate, and meaningful numbers of patients appear to be cured. So that was this beautiful journey. And how does that relate to vaccines? Well, now we actually have patients with really good immune responses who are deriving clear clinical benefit. So now you can interrogate those patients and say, well, what is it that's happening in you that is clearly killing your tumor? And we now had the tools to be able to sequence their tumors, analyze T-cell responses. And so this famous paper in the New England Journal at the end of 2014 suggested that it was these mutated so-called neoantigens that were driving the immune responses that were leading to cure. And so that spurred 4 companies to form or to focus. So Gritstone, which obviously I started, Neon, which was a third rock company with Kathy Wu and Nir Hacohen are some of the key co-founders out of academia. And then 2 other companies you've probably heard of, Moderna and BioNTech, have been around for 5 years or so by this time, but they found an application that made a lot of sense because you could quickly make personalized neoantigen vaccines using mRNA technologies. So the 4 of us really sort of then really started diving into the field. And it's been a journey since then where gradually we've started to figure out how these products can work, where they work best, and how to think about regulatory approvals and so on. So it's been a long journey, but I think we're slowly getting there. This is the classic, innovation life cycle, right? You have 2015, all of us got funded real easy, then it got really hard, and now we're getting back to a point where I think we're going to start to harvest some of the fruits of all that work. And first up will probably be Moderna with their Merck collaboration, where they've got these phase 3 programs now running in melanoma and lung cancer, all in the adjuvant setting, because it seems that's where these drugs are going to work best.
So obviously we've been waiting a while to cure cancer.
Yeah.
You guys need to get cooking on that. What do you think is most promising in terms of new therapies and ways to use tech to potentially target oncology?
Yeah, so it's a I think a multidimensional answer. I think we all know early diagnosis is key. And so that will require us to use diagnostics in a clever way. And there are smart people working on this. And the problem, if you think about it, is that once you've got an established tumor, I've got usually lots of mutations, and the tumor's usually reasonably voluminous, right? There's quite a lot of tumor cells around, and they spill out their mutant DNA into the blood. And of course, I can now detect mutant DNA. And so that has become a big enterprise, and companies, Guardant and obviously Foundation Medicine picked up on this, Natera and so on. Other companies are focusing on this notion of sequencing the mutant DNA in blood. And if you find mutant DNA, obviously you're highly confident that there's a tumor there. And so this has become a kind of staple of early diagnosis. The problem is, if you don't know that there's a tumor there, you don't know what mutation you're looking for. And that's obviously a fundamental kind of chicken and egg problem. And so you can't do the things we do to monitor tumors, where you sequence the tumor, identify the mutations, and track those in blood. That doesn't work if you haven't got a tumor, right? You're trying to do early detection. And so what you're looking for are nothing patient specific. It has to be a pattern that is commonly found in early tumors. And you've got to find it in an analyte that is present in the blood at a reasonable level. because mutant DNA is often, particularly in early disease, there might be one copy per 10 ml of blood. So if I do a 5 ml blood draw, I may just miss it because, you know, I got the wrong 5 ml of blood. This is a real problem when you're looking for these single rare analytes. So you've got to go for something that is more abundantly present, and ideally something where you're looking for patterns. And this is something that Grail did a lot of nice work on. Guardant's working on this. So people are looking for these epigenetic patterns where you're not looking for single mutations, you're looking for alterations in the epigenome, right? Altered methylation, whether that's DNA methylation, histone methylation, these changes in patterns of epigenetics that are seen system-wide across the genome. And you can look for like 20,000 alterations. And all you need to do is find, you know, maybe it's 100 of them. It varies, obviously, a little bit. The specifics vary, but I need to find 100 of the 20,000 I'm looking for. And if they all point in a certain direction, then I'm pretty confident you've got cancer. So this is a tour de force of sort of diagnostic technology. I need sensitive and very robust assays for these combination analytes. And then I need informatics to be able to say this constellation of genomic features or epigenomic features means you've got a very high probability of having cancer.
Interesting. I mean, actually, no, I'm curious to get your thoughts. So obviously there's like the machine learning and like all the data science component side of the problem, which I would say is like pure tech. Where's the constraint on the bio side?
So there's what is the definition of a tumor is sort of a piece of it, right? And figuring that out. So you've just got to collect enough data to be able to confidently say this pattern of markers equals high probability of cancer. That's the biology piece, right? The tech will figure out, or the molecular diagnostic, you know, the engineering will figure out the way to measure and the way to measure robustly and sensitively and with adequate specificity, et cetera, et cetera. So that'll give you the way to analyze blood. Putting it together on the biology side, you've got to collect the samples and annotate them appropriately from large numbers of subjects with a variety of tumors and non-tumor to be able to correctly define or put in place your definitions of good versus bad, of high risk versus low risk, etc. And then obviously all of that is done on a per patient basis using sort of high throughput sequencing and bioinformatics. So that's kind of the piece, and that will get you to a world of early diagnosis. So that's a part of the puzzle, right? Because that clearly is necessary if we're really going to move the needle on reducing the number of people dying from cancer.
Speaking on bioinformatics, it seems like what you're doing with Peptone is very AI drug discovery based. Would love to hear more about how you're thinking about using these technologies.
Yeah, so early detection is key, and then the treatment, obviously early treatment is key, and that pretty much any drug you want to give, the earlier you give it, the better your chances of eradicating disease. And that's certainly true of immunotherapy. And so that's a reason the vaccines are being used in these early stages, right? In the adjuvant or neoadjuvant stage, because you've got low volume disease. The disease has not yet developed a whole bunch of therapy resistance mechanisms. And usually it's genetically relatively simple, and they just get more genetically and therefore immunologically complex the longer time passes. So treating early is always good, but we obviously need really good drugs. And so we're continuously hunting for better drugs that sort of hit the foundation of tumor biology. So that leads into the company I'm now working with called Peptone. And Peptone has developed a technology that is being used, seems to be useful to develop drugs against targets that we all wanted to drug, but historically have been really hard to get a grip of. And these are things like transcription factors. And the problem with transcription factors is that they have particular patterns of charge because, of course, their job is to bind to DNA and recruit other proteins. And so they often have a fairly highly charged environment, and that makes them energetically relatively unstable. And what that means is that they don't tend to form crystals. So we don't have crystal structures of these really attractive drug targets. And therefore, as a class, we've really struggled to make drugs against them, even though they're foundational to biology and they're things we really want to hit. How do we tackle this problem? Well, we've seen these amazing innovations in the world of drug discovery using artificial intelligence to identify structures, right? And so, you know, the success of AlphaFold, the Nobel Prizes awarded last year to three, you know, inspiring characters who've built these beautiful models for protein structure prediction, right? So all you know is the primary amino acid sequence, meaning the string of amino acids that are joined together. And what they've been able to figure out is how that string will fold together and create this complex three-dimensional structure, which is, of course, what we actually make drugs against. Now, it's beautiful work, but it has limits because you can only predict on structures that you've trained on. So in other words, ordered proteins where we have crystals that we've been able to do X-ray crystallography on have been the bread and butter for generating training data for this, AlphaFold, let's call it that, generically. There's a version Boltz, which is a public domain version. There's lots of flavors of this, as you know. But they all have the same feature, which is they've trained on crystal structures, knowing the amino acid sequence, and they've learned how amino acid sequences do fold together based on all of these training data. We've learned the basic rules, and there's a lot of elegant sort of biophysics going into this, but that's really the kind of at the heart of this. Now, the problem with things like transcription factors, they don't form crystals, so we have no training data. So if you plug one of these proteins into AlphaFold, it'll give you, 50 low confidence predictions. It'll say, look, here are 50 very different structures. One of these might be one of the right structures, but I don't really know. The confidence for all of them is low. And that obviously doesn't help you discover drugs, clearly. So how do you solve that? Well, traditionally, when we think about, hardcore protein structures, we thought about crystallography. That doesn't work, as I say, for these disordered proteins. And just parenthetically, they're given the term disorder, which is a little bit misleading because probably these proteins just have multiple viable structures. And so they can adopt different structures and they flip-flop between a variety, which means they won't crystallize into one energetically stable structure, but they have multiple, structures, all of similar energy states, none of which is particularly preferred. And that's why they constantly are changing their conformation. And so druggable pockets form and unform. It's a complicated milieu, and it doesn't lend itself well to our sort of static modeling that we're so used to. So nuclear magnetic resonance has often been developed, but it doesn't work for big proteins, and it has additional sort of technical limitations for solving these kinds of problems. The team at Peptone, led by Camille Tamiola, the CEO, he's a biophysicist, and he worked at Chris Dobson's lab in Cambridge and then in Groningen. And he, he worked on NMR, right? So he really understands that technology, but could just see the limitations that we had when we're trying to tackle transcription factors. So an alternative approach is a thing called hydrogen deuterium exchange mass spectrometry, HDXMS. Big name, I'll call it HDXMS. The principle is reasonably simple, right? Which is mass spec is where you fragment proteins and you measure the fragments, and the time of flight depends upon their size and their charge, right? So take a protein, fragment it, you'll get a spectrogram that reflects the fragments that are generated. So far, so good. If you then label that protein with deuterium, what does deuterium do? It exchanges for hydrogen atoms, and it doesn't change charge. It doesn't change anything apart from the mass, because you've got that extra neutron. So if you then do the mass spec analysis of that labeled protein, you'll get a different spectrogram because obviously the fragments are a little bit heavier. So I can see the difference between the labeled and the unlabeled. Now, what people traditionally have done with this technology is to say things like, if I've got an antibody that binds to a protein, and I do that, I put the antibody onto my protein, I then do the deuterium labeling, deuterium won't exchange underneath the regions where the antibody is binding. So effectively, I get a picture of the footprint of the antibody. Does that make sense?
I see. And from there, you're using that footprint
to figure out where the antibody is, by where the actual, what's called the epitope of the antibody is, the bit that the antibody actually binds to. So it gives you this beautiful understanding of literally where the antibody is binding without having to make a crystal. So that's what it's been used for. Now, how do I deploy that against these disordered proteins? If you just sort of do mass spec with deuterium exchange on a standard protein, the first time point that you normally can get in an experiment is about 1 second. Well, all of the deuterium exchange happens inside a second, so you don't really learn anything. So the innovation that Camille and his team have pioneered is to do the hydrogen deuterium exchange in millisecond timescale. And what that means is that if you perfuse the protein with deuterium and you arrest the reaction within a few milliseconds and then do a snapshot mass spec experiment.
You have like higher resolution of all the...
Exactly. What you'll see is the surface hydrogen atoms will exchange. But any hydrogen atoms buried down deep in a cleft or a pocket won't exchange that quickly. It takes longer for the deuterium to reach down and get into the pockets. So you get effectively, over time, you get a map of the surface topography of the protein.
Do you get a map of what's beneath as well?
Here's the clever bit. You now go back to AlphaFold or some version thereof, and you say, okay, you've given me 50 low confidence predictions for what the structure of this protein might be. I now have data. I've got experimental data that tells me this is the way in which deuterium will diffuse into this protein. Do any of these predicted conformations fit with my observed experimental data? And guess what? Often,
got it.
One of them will fit perfectly.
So you're kind of like interpolating from that.
You're triangulating between these two totally orthogonal data sets, right? One is a prediction, the other is data. And you end up with what looks like something that fits perfectly. So the prediction fits your experimental data. Now you've, right, isn't that cool? So now you've got your teeth into something that looks real. The HDXMS data also tells you about, accessibility of pockets. So now we say, all right, well, let's do some in silico medchem, right? The classic virtual screen. Virtual screening has a bit of a mixed rep because sometimes it doesn't work very well. But here, we've obviously got this very nice model of where the pockets are, what they look like, what the specific atoms within the pockets are. And we can then do virtual screens to find small molecules that coordinate appropriately.
I know virtual screening has like a pretty mixed reputation.
Yeah.
To what extent do you think it's just a bad approach versus the underlying data that we have isn't high quality enough yet, and that being the constraint?
Yeah, I think obviously more training data, better resolution training data will always help. So I think that certainly, I think that the utility depends on the application, right? If you're simply taking a protein where we've already got a crystal and you're saying, I'm going to apply this for now virtual screens. Well, frankly, you're not helping that much because we can already do that, right? With, high throughput screens using traditional med chem, right? We can do structure-based drug design and so on and so forth. So I think that's just not the right application for it. Trying to effectively get to the same destination with a different path doesn't make a lot of sense. I'm more excited.
Speaking of destination, how did you get to this? Like, walk me through very early Andrew days. You're 14 years old. How do you end up here?
I was always fascinated by medicine. I just loved this notion of understanding disease and treating disease. And I was that kid that sort of used to buy, back in the day, you could buy things like pig's hearts from a butcher. And I'd take them home and start dissecting them with my little sort of amateur dissecting kit. And I had no idea really what I was doing. And you'd have a little book next to you and be like, okay, this is a thing called the mitral valve. And, you're sort of just figuring it all out. So that's kind
I still remember the smell of them.
Yeah, they are pretty stinky, right?
Yeah.
They do smell of pig. There's no getting away from it. And then you do the same with the eyeball, all that kind of stuff. So that was my sort of passion. And I guess I just followed through with that. And when it came to going to university, in England, the system's a little bit different from the US, where we specialize pretty early. So, at the age of 15, 16, you start to pick your specialization, right? We do a small number of exams from the sort of 15 to 18 range called A levels. So I was focused on, maths, chemistry, biology. That was my sort of shtick. And then I applied to med school, got into med school, and so that's it, right? Your path is set. We don't have this lovely. Cambridge does it differently. I went to Oxford, where we're kind of on that journey. Cambridge does this thing where they still try and keep you as a little bit of a generalist in your undergrad time. I like that model. So that's maybe a good thing, because you'll just maintain that breadth, because I think it serves us all well to sort of have some understanding of the other aspects of life, right? Because clearly philosophy plays into science pretty heavily, and these days, obviously, without opening that whole can of worms, an understanding of public appreciation of science is important, and you won't have that necessarily if you've been so myopically focused on just pure science and working with pure scientists for years. Anyway, so that was kind of my journey into this. So back to peptone. So the peptone thing now, what's so clever about it is you make a small molecule to these disordered protein pockets, and then you run another one of those HDXMS experiments, and if the molecule binds, it will protect those residues from deuteronation. And so you can immediately confirm that your small molecule is binding where you wanted it to bind. So all of that can happen in a handful of weeks. So you literally go from starting structural work with a recombinant protein to having binding small molecules that bind into a unique pocket that no one's ever seen before in a few weeks.
Wow.
And if you've got a functional assay, you can just throw it in, and it won't be a very potent molecule, obviously, but you'll start to see it inhibiting in a functional assay. And this is all, again, it's in a matter of weeks, low expense and weeks. And so that's what, to me, that's what's so exciting about this, because you can tackle proteins that everybody wants to tackle, but frankly, we've never really been able to tackle before. So I'll give you a good example, c-MYC, right? c-MYC is an oncogene that pretty much anybody who's been working in oncology for a while has tried c-MYC, and no one's succeeded yet, right? The most advanced assets we have made it to phase 2 and then failed. And there's always something new trying for c-MYC, but we're in this totally different part of c-MYC, which is totally disordered. So like many proteins, it has regions of order, regions of disorder. The disordered regions is where we're focused, obviously. And so now we've got nice binders that seem to functionally inhibit, and maybe those will turn into drugs.
Doing this in a span of a few weeks sounds very software-like. I mean, biology in general takes years and years of research before you even reach something useful, and then many, many years more in order to go through the FDA process. What's allowing things to happen so fast now?
If you think about the traditional way we've done drug discovery, and I spent some time in big pharma, right? This is, well, it's true of little companies as well. Little companies maybe move a bit faster, but essentially you start off saying, what's a good target? So you have a whole group working on target validation. And when you've got a new target, let's say for cancer, the subject I know best, you'll say, I think that this gene creates a protein that is important to a cancer cell, and if I inhibit the function of that protein, I will inhibit the growth of the cancer cell and thus treat the patient, right? That's the sort of logical flow. So how do I get information on that? Well, I don't have a drug, right, by definition. This is a target discovery campaign. So I'm looking for kind of circumstantial evidence. The best probably are the knockouts, right? So you say, do I have a knockout of this protein, and does that seem to do anything to cancer? And occasionally the answer is, yeah, we actually do have knockouts, and they either do get cancer, right? So an activating mutation.
So this is like a BRCA4 type.
Yeah, thing. Like BRCA would be a good example. Like KRAS is a good example, right? So KRAS is mutated in many solid tumors, right? Pretty much every pancreatic cancer and third or so of lung and colorectal cancer has a KRAS mutation. So that obviously is strongly supportive evidence that KRAS is an oncogene. And then you do transfection experiments and you put activated KRAS into cells, and guess what? They start growing proliferatively, uncontrolledly, and it starts to look like a tumor. And then you do a transgenic mouse where you put the activated gene in, and Dave Tuveson did this in the so-called KPC mouse, where you put in 3 mutations, one of them being KRAS, and then guess what? That mouse gets tumors in their pancreas, right? So now this is the body of evidence you build to say, KRAS is a therapeutic target, let's make a drug, right? That's kind of the approach. Then you have like 3 groups going to some committee that's saying, we want to begin a drug discovery campaign against our target. And the 3 groups will compete, because usually there's finite resources, and maybe one of them gets blessed. So they begin the campaign. So what do they do next? Now they've got to make some cell lines or some assay systems that use that particular target protein and enable you to do high throughput drug discovery, right? So obviously if it's an antibody, you're panning and screening. If it's a small molecule, you're doing HTS assays, high throughput format with chemical libraries. So you've got to get those assays working. That's a bunch of work, right? So that's 6 months of work. Then you start the screen, and over the next 6 months, hopefully you find small molecules, let's say, and you find things which are hitting in your assay. And then you show that that works in a cellular system, and now I've got a hit. That whole process can take 18, 24 months, spend a bunch of money. Now I've got a hit. Now I've got to optimize that hit to try and turn it into something that looks like a real drug. That's called the hit to lead phase. That takes more time, more money. So that's the traditional path. It's all very rational and reasonable, and we've been scaling, but we've been scaling really on the chemistry piece of it, the high throughput assays, robots, all that good stuff.
Yeah.
What's different now, perhaps, using the approach that Peptone is developing, is that you compress all of that work into a few months, because we're working on targets that frankly have already been validated, but we're working on regions of those targets that nobody's been able to drug before. I mentioned c-MYC. There's a famous protein that causes Tregs to form. People are excited about potentially hitting that as a cancer therapeutic. But No one can touch it. It's a transcription factor, so we don't have anything, but it's a great target, right? Everybody wants to hit that. Lots of circumstantial evidence that tells you it's a good target. So what we do is we take those well-validated targets, we just need the recombinant protein, and then we do the HDXMS work. That's obviously a one-day experiment. Then there's some analytics behind it. Then you do the virtual screens. That's obviously, again, you're limited by GPU access, and so that's obviously a piece of the company build. It's getting access to GPUs at scale at a good price. You've done your virtual screen. You then order those small molecules from any CRO, right? You go to Evotec or Enamine or Molport or whoever, buy the small molecules, bring them in, and all you need now is to do the HDXMS experiment again. Some of them will bind in that assay, and now within the space of 3 or 4 weeks, I've got binders into my novel pocket. And then if I've got a functional assay...
And you have a supply chain that is very efficient and
It's
fast.
straightforward. These are small molecules,
Yeah.
right? So that's how and why you're able to move this quickly. And so it's exciting. Now, again, we're not doing sort of de novo target validation work. We're taking that as done because we're working on things that people have already been focused on for years. Our lead campaign is actually against the androgen receptor for prostate cancer. And people have been focused on that for 20 years, well, longer actually. I mean, surgical castration, which obviously reduces androgens, was the first treatment that was developed in the 50s. Charles Sawyer's obviously famously developed novel AR antagonists, which sort of birthed enzalutamide and medivation and so on. They all hit the androgen receptor, but they all hit it at the end of the receptor where testosterone binds. They compete with testosterone. And the challenge is that the tumor cell develops resistance mechanisms, and they either mutate the binding site so the drug can't bind, or they amplify the androgen receptor so it outcompetes the drug, or they splice out the ligand binding domain so that the drugs have lost their docking site, so they can't work because the whole region is gone. But the bit of the receptor that's always present is the N-terminal domain, the bit that actually recruits the activation, the transcriptional activators onto the DNA. And that's fully disordered. So we focus on the N-terminal domain of the androgen receptor, which people have wanted to drug, but no one's successfully drugged before. And so we don't have to do work convincing people that the androgen receptor is a good target. Everyone knows it's a great target. We've got approved drugs. We're just tackling it in a unique way. And we're doing it in an efficient, fast, compelling way that in principle may give us drugs which will be active in all the settings where today current drugs are inactive. That's what's kind of cool about this.
So how do you think about this from the lens of the whole platform versus asset kind of continuum and that whole side of things?
In our industry right now, of course, we're in this desperate time where capital is so limiting, and the eternal debate is platform versus pipeline. And the simplistic approach is say, look, just give me a single asset. I can probably develop that somewhat virtually, highly capital efficient. Obviously, the problem is there aren't that many assets out there which are frankly good enough to merit that kind of focus, right? Because if you take a novel asset against an old target, then chances are it's not going to be that interesting. And if you're going after a new target, then everyone's nervous that it's just not going to work. And this is a risk-off environment where people, even if you're going to do it efficiently, they don't want to put all their money into a single asset bet because it's binary. And it's still not that cheap to develop a drug and take it through to proof of concept in humans. The pipeline of interesting assets which can be developed as an asset-only play is shrinking, and by definition, innovation is thinning out. And yet we all know innovation is what really creates value in our industry. So then it inevitably, inexorably draws you back to pipeline, but then pipeline can be expensive and people don't want to spend a ton of money on platform development. So
the very large acquisitions generally tend to be platform acquisitions.
Right.
Given that's the case, why is it that there's not enough resources going towards that?
They're just, many platforms are expensive, and as we've got into novel biologics, they can get really expensive. And obviously, I know this. At Gridstone, we built this very bespoke platform for making patient-specific vaccines. It was perfect for purpose. It was as efficient as we could make it, but nonetheless, it's capital intensive. Right now, the question is, can we develop platforms which are innovative, will act as a flywheel for high-quality, innovative drugs against high-value targets? But can we do that in a capital efficient manner? And I think the answer is you can, but you have to pick your platform. And obviously That's what we've been trying to do at Peptone, is stick to a highly capital efficient, highly scalable platform that does act as a flywheel for high quality, novel assets against high value but novel targets.
You have an interesting skill set, obviously, at the intersection of extremely deep knowledge on bio, but also a lot of knowledge on like the commercialization, fundraising, and that whole nightmare side of things. Were you always interested in the business side, or was that something you kind of had to learn to get these therapies out to market?
No, I'm a sort of generalist, really. So, I like the breadth, and of course I like the business side of what we do. I spent a few years at McKinsey, which was really good training to sort of think about fundamental strategic questions, and obviously trying to develop strategies for drug discovery and drug development. And it's hard. I mean, that's the thing I love about our industry, infuriating though it can be. We're trying to do something that ultimately is so rewarding, right? I mean, to help patients who really need new therapies is as good as it gets. But we're doing it in a way which is really intellectually challenging and demanding, and it calls upon everything you know. And you will never know enough to be able to do this on your own. You're always in a team. I like working in teams. I like working with people super smart. I love that sort of creative debate where you just throw issues around, and there's no inherent winner. It's just about, let's try and find the best idea, and let's just bat those things around. That's what I really, really love doing. And I've done that. That was true at Gridstone. It's been true at Peptone. These are just complicated problems. Nobody has the answer all to themselves. You just have to collectively try and frame the problem correctly, accurately. And that takes different dimensions of input, right? Because a problem, there is a financial aspect to every problem, right? It's no good having a solution if it cannot be funded.
Yeah.
And that's what makes our industry so hard, is that these, what's fundable today may not be fundable in 10 years. I mean, that was the Gridstone experience, right? It was 9 years from start, when funding was abundantly available, to a point where we couldn't raise any more capital. And it wasn't a failure of the science. I mean, the science didn't deliver, barnstorming efficacy as quickly as we would have liked. But that doesn't mean it doesn't work. It means you're learning. And so we've learned about where those products are working. Of course, that's natural and normal.
So what do we kind of do about this? Because obviously, this is the single most important field to be investing in from a pure impact standpoint. Investors are always going to be financially driven. What needs to change about the biotech industry as a whole in order to make it more investable? Is that better technology to make things move faster? Is that virtual clinical trials? Like, how do we actually make this an industry where investors are incredibly excited to invest in the same way they may be about, B2B SaaS or whatever it may be?
Yeah, that's a really good question. So I think it's ultimately down to time and cost, right? What you want is, you want to be able to innovate and deliver critical proof of concept data quickly and as cheaply as possible, right? That's ultimately what you want to do. And in tech, you can do that in the space of months. We can't do that in biotech. It takes years.
Well, that's contrary to what you said earlier about now being able to find initial version.
Well, that's the initial version. Then you've got to get to the clinic, right? So again, it's not enough just to have a great drug and be perfect in monkeys or mice. You've got to get through clinical trials. So it's always going to take years, plural, but it doesn't have to take 15 years. It doesn't have to take 10 years. So how can I make it take, 5 to 8 years and do it in a manner that doesn't cost billions of dollars, but just costs tens of millions of dollars?
Yeah.
That's kind of the credible challenge. And so this is where tech obviously intersects so beautifully with the biology, that we learn, we train, and we deploy, but we just have to do it in really thoughtful ways, where you're trying to minimize risk and time and cost at every step of the value chain. And of course, the tragedy is that at some level, the tech most easily fits into the cheapest, fastest bit of the value chain, which is the preclinical stuff, right?
Yeah.
The drug discovery stuff. That is not the slow and expensive bit. The slow and expensive bit is the phase 3 clinical trial. So how do I improve on that? It's a whole different question, and tech, I think, does have a part to play there. It's something to do with the organization. It's the way we structure our trials. It's the way we pay for them. I mean, to be honest, the cost of running a clinical trial is massive, and that is something that we don't control. We as a biotech industry, we don't control that, right? That's about data collection, veracity of data, and paying hospitals to collect data and to treat patients. And some of those elements are actually pretty compressible.
Yeah.
And there are people working on that, and that's really important. And you want all of that to come together. And we want to be smarter about the trials we run. You know, this is one of the things I like about patient selection, is that are able to identify preclinically and then clinically that often it's a subset of patients that's going to benefit from your drug. And as we get smarter, we get better at knowing what that subset is. And we run into this challenge in the clinic where people say, oh, I can't do a trial if I'm selecting for just 20% of the population. Because that's I have to screen 10 to find 2. It's inefficient.
Yeah.
My nurses don't like doing that. And so you run into this very kind of prosaic challenge. The patient wants the best drug. So you've got this intermediary where the economics for them don't make sense. And so we have the incentive to solve that problem. And I think increasingly we're getting the tools to solve the problem, where we just have to start automating more and more of this process. And this is all about doing broad screening of everybody in the healthcare system and capturing their information in some centralized way,
and having virtual patients of some sort.
And having access to that, so that when you're trying to run a clinical trial, you don't have to run around trying to, find the site that's prepared to screen, to find the patient. You just know the patients are there. And you can go to the site and say, your database tells us that you have these patients who have the disease that we think we have a drug. We're going to get there. But I've been working on this for 20 years and we're making progress. It's slow getting there, but that's obviously the way we start to change some of these downstream costs, which are, those are the biggest costs.
So say in a hyper idealistic scenario, let's say it's as fast as what software was, a few years ago.
Yep.
If we get to that point, I think it's an incredibly exciting asset class to be investing in, not just because of the impact, but also financially, it's really exciting as well to investors who may have been scared away from this field. How do you invest in the actual acceleration of, so not just not the platform, not the pipeline, but the underlying sort of technology necessary to make the whole process faster?
Yeah, I mean, there are companies obviously trying to solve each part of that, of the value chain. And they meet with varying degrees of success because there's a lot of entrenched forces in our clinical research environment. And again, this is not what I do day to day, but this is where I will end up, I hope, soon again. And I've been in this world before. You're doing phase 3 trials. They're going to take you 3 or 4 years, and they're going to cost you $100 million plus, right? And if you have to do several of these, which sometimes you do, this is where the real cost racks up. So again, if you want to impact our cost curve, this is where you're going to have the greatest impact. But it's hard because you've got a lot of rent seekers living in that space. And we need to disintermediate them if we're going to kind of change the timeline and the economics.
If anything happens to you soon, we know why.
Yeah, right. I mean, we all know this, right? And there are companies who try to get in and disintermediate, but it's hard. But the incentive is there. So I haven't given up because I think ultimately everybody, unfortunately, is going to be a patient. And everybody needs to step away from their own day-to-day economic, whatever their job is, and just think about the family or themselves who are going to be a patient, where all of the other dimensions fall away and all you want is the best care for that person.
Yeah.
And that means you want the right drug for you, whatever the disease. Obviously, I work mostly in cancer, but it's true of other diseases too, which means you want people to figure out that, not all hypertension is the same. Some people have hypertension caused by this, others by this. What is the best drug for you? Hypercholesterolemia, we're starting to get into this, right? Asthma, look at this, right? That's traditionally a sort of field that has not had a ton of patient segmentation, but we're now thinking about eosinophil count as a way of segmenting patients because we've got therapeutics that do and don't work depending upon your eosinophil count. That's true of COPD as well.
Yeah, not to mention all the unique sort of like overlaps of these different diseases, and
right,
and every person really is super unique, even if they have the exact same disease.
Right, and so eosinophil count is super beautiful to work with because it comes off of a regular CBC. And so that means a physician doesn't have to have the thought, oh, let me measure the patient's eosinophil count to see if they're eligible for this particular drug, because they're going to have the data. It's going to be in the system. And so I can just go into the database and find patients who've got two things, right? They've got a diagnosis of COPD and an eosinophil count of over 0.5 or whatever your number is. And it all happens for me magically. In oncology, we run into this problem that it's complicated, and there's people looking at lots of different attributes. So you've got colorectal cancer. Okay, well, do you have a KRAS mutation? All right, well, do you have a BRAF mutation? Do you have a BRCA mutation? Do you have, are you the MSI high phenotype? I mean, the numbers of diagnostic tests starts to stack up. And generally, particularly in sort of the community, physicians just don't have the bandwidth to think about all of these things and test for all of these things. And so the notion of patient selection and segmentation just starts to fall away before it's even got going. That's the bit, I think, where we can change, first of all, which is to start routinely testing patients. And that is starting to happen, right? Guardant, Tempus, these are companies that do broad assessments of patients.
It's so interesting because it kind of comes full circle where you're saying the most important sort of low-hanging fruit for cancer is early detection. The most important thing for early detection is obviously like high quality diagnostics, as close to real time as possible, as frequent as possible. But then the most important thing for speeding up the life cycle of creating therapies quicker seems to be having this really high quality data set of patients. And so in a weird way, the early detection also helps with the cure or treatment of disease, because it provides us with the data set necessary to go through clinical trials quicker.
Yep. Yeah, totally. Yeah. You're basically sort of documenting, annotating a patient's disease comprehensively at the outset is really going to help with everything. It's going to help that patient. It's going to help with diagnosis, detection, and treatment. Yeah, so I totally agree. And, that can be done, right? There's actually not much stopping us apart from just simple economics and habit right now, right? But those can change, right? The economics can shift because you've just got to demonstrate that it's worth it. And I think generally that's true, that you will be able to connect the dots and say diagnosing these patients accurately will lead to better outcomes. And there's obviously lots of data that sort of get in that direction. It's not often done at an integrated healthcare system level. It's done for an individual drug, and you can aggregate that in a clunky kind of combination analysis. But better will be when systems start to do this. And, national health systems can do this. Big integrated groups like Kaiser can do this. So I think it's going to come, and we'll all be better off for it. And therefore, let's keep driving to that future.
That's exciting. I think the number one call to action for investors would probably be just keep investing in the space. And one of the issues I've noticed is that the sort of consistency of capital in biotech is very like boom or bust. And that doesn't lead to good research, because you need to know that you're going to have future investment and that you can continue to do the research. What would be the sort of call to action you would have to, a young bioengineer or biophysicist out there that's trying to make the biggest impact on this field? What advice would you give them?
Yeah, I mean, define the opportunity that you think is exciting and moves the needle, because I still cling to the belief that innovation that helps patients will ultimately be paid for, right, and will create real value. So that, I think that's kind of the first step, is like really focus on something that makes a difference, that is practicable. And so I think you've got to.
It's crazy that statement is one that is controversial.
Yeah, we sort of, the boom and bust has a consequence, right? The consequence is that during the boom times, ideas which are maybe a little bit far-fetched or, oblique can get funded. And of course, what that often means then is that they don't succeed, and the people who invested in those then get bitten, and then they retreat. And so you create, the cyclical nature of our financing environment is a little bit self-perpetuating, right? That when the boom comes, because right now we're so starved and really good companies and good assets are probably substantially undervalued. When the boom comes, it's going to be a good boom, because valuations will rise appropriately and people will be rewarded for the good work they've been doing. So that's great. But alongside that, we'll get the froth, and then stuff that doesn't deserve to get invested in will be invested in, and you're sowing the seeds for the next crash, right? So we just have this inherent cyclicality, and we always talk about generalists versus specialists, right? And the specialist investors are the ones that in principle really understand the space and can invest smartly. The generalists often will follow the specialists, but in principle that's the way it should work, right? Generalist money follows specialist money. Unfortunately, what you sometimes find is that it flips, and you find the specialists thinking
where's the money going to be? Therefore,
the share price will rise when the generalist moves into this stock. So I've got to predict what it is the generalist is going to like, and I'm going to invest accordingly. And you end up with the generalist driving the specialist for an understandable reason, but one that at some level is flawed. And we see that phenomenon kind of playing out as well. So
how do you align sort of collaboration and investing between the generalists and the specialists? Because I think there is some logic to the fact that if you're a specialist and you want the companies you're investing in to succeed, you need to ensure that there's a relatively high likelihood that they're going to secure additional financing. And given the vast majority of people are not specialists, the vast majority of capital is controlled by generalists. So how do you ensure that you have that specialist expertise deciding where capital is allocated while also ensuring that you receive additional sort of follow-on capital from the generalists?
Yeah, I think the answer is discipline. I think, investment discipline is the answer, because, I know really good investors who are just can do this. So I think the answer is, if you're a specialist, you're going to be investing early when the situation is unclear to a generalist, right? They can't really understand what is this drug doing? Why is it going to work? How can it work? And the specialist will understand that. And so that's the space for the specialist, right? Who can understand the logic and can, in a disciplined fashion, due diligence, and then invest in good ideas, trying to solve good problems with good science. When I talk to investors like that, we have a rich conversation where I'll be talking about, protein structure and the N-terminal domain of the androgen receptor and the recruitment of transactivators and how splice variants of the androgen receptor arise. You can have that conversation. Everybody understands what you're talking about. If you try and have that conversation with a generalist, they don't know what you're talking about. And so you end up, I've had some really awkward conversations with generalists who've wanted to or been interested in coming in early, where the science is rich, and they ask you to talk about it in terms of, can you give me a Star Wars metaphor? And it's like, it's pretty hard to explain, T-cell receptor binding to an HLA peptide complex with the Star Wars metaphor, right? There's just a fundamental mismatch. And so I think generalists should somewhat stay out of the early game, because it's just not a safe place for them to invest, because they don't have the knowledge. They're smart people, it's just they're not expert in this particular domain. This is where the specialist should be. And then once the asset moves to the clinic and you start to get clinical data, everybody can understand clinical data. Show them a clinical trial where you've got a randomization and the control group, gets lots of disease exacerbation and the test group does not. Everybody understands that. That's the point at which generalists can appropriately invest. So if you keep that discipline, and if the value creation ladder mirrors de-risking and clinical data, then you have a healthy system. And that's obviously what we sometimes have. But then we tend to swing either side of it. But I think that's the answer, is just keep that discipline. We all stay in the swim lanes for which we're kind of optimized. And then the ecosystem hums along pretty nicely. It's when we get out of our swim lanes and we're suddenly doing things we shouldn't be doing, then we can get into trouble.
Interesting. Yeah, I mean, I have some thoughts on that, which might push back a little bit. So one, I noticed that specialists tend to be such experts in their field that they can be jaded or almost have a level of pessimism that might not allow for like some of the more out there ideas to initially start, or these initial experiments to run, especially if they've seen past failures in a specific approach. So maybe in some regards, the generalists' lack of knowledge might mean that, more people get started with experiments that have a low likelihood of working but could be interesting. I would love to see more large downstream specialist capital. So I think one of the issues is that most specialists just don't have enough capital to be able to, lead large rounds or really move the needle on the capital markets side of things. And so potentially on having generalists writing, small early checks, even if the precision is low, might mean that more scientists get to try things. But at the same time, having more Wall Street investment bankers with massive balance sheets that just so happen to have, bio PhDs might be a good thing for the industry. But I do think that unless you get both generalist and specialist capital in a company, it's hard to sort of have that steady sort of flow of capital necessary to keep doing research and to keep building things.
Yeah, I think So I think your argument is you want abundant specialist capital. Well, I'm not going to disagree with that. Obviously, I don't control that. And the way that will happen is because they'll make disproportionate returns and people will then give them the money, right? So I think it behoves specialists to deliver outstanding performance, and thus they will attract greater capital and their funds expand. It's tricky, I think, because there are finite numbers of great ideas. And, there's this whole debate within venture capital firms about how big should they become. Because, a successful VC, the temptation is always there to grow bigger, raise more money, have bigger funds, hire more associates and so on. But probably, and I'm not a VC, so I'm over my skis here, but probably there is some kind of maximum within a VC firm where you've got the right partnership size, that every idea is appropriately vetted, and there's probably too big, where somehow the system kind of breaks down and you end up with like inappropriate committee making, decision making, or you sort of fragment and then you lose the benefits of partnership. So I think there may be some sort of natural constraints on how much capital can live in a single fund. You should talk to a fund manager about that. Interesting question. Your first point about, sometimes specialists can become jaded. I think that's true, but I don't think the answer necessarily is to say, so we should bring in enthusiastic but uninformed capital because they might sort of fund something that could succeed. I mean, that's true, but that's crazy inefficient. It's probably going to lead to a lot of value destruction rather than real value creation. And yes, the lucky person who doesn't really understand what they're investing in, but puts money in and it takes off, will be happy. But then the 20 other people who did the same thing and all failed will not be so happy. So I'm not sure that's the answer. I think the answer is specialists need to do a better job and don't be jaded, right? Invest in innovation and keep doing your job, which is be smart and invest in good ideas that solve clever, important problems in clever ways.
Yeah, I generally agree with that. So, yeah, I mean, super exciting the work that you're doing with Peptone. I feel like your career arc has just been incredibly both impressive and impactful. The amount of capital and energy that you've been able to put towards like these really groundbreaking therapies is something that I really want to see more of. I'm curious if you have advice for me as someone who, is a relatively generalist investor, but still, I like to go deep on a bunch of different subjects, whether it's biotech or autonomy, or even recently, learning a bit more about material science. Where do you think are the best places for me to look in terms of where to invest? Is it the research labs, the church lab spin-outs, or is it the independent scientists scattered everywhere across the world doing cool stuff? Like, what's my job in basically ensuring that these companies exist and have the best likelihood of success?
So right now, my sense is, obviously, given the low valuations across biotech, that you can probably make good money. And again, I'm not an investor, so this is caveated heavily, but there are probably some very good investment opportunities in public markets right now. And I know obviously a lot of investors are looking at various public opportunities because, you're proximal to data events and you have liquidity.
I only do private market, ultra early stage.
Okay.
First check.
Okay. There you go. Yeah. Because if you're a sort of crossover investor right now, again, public markets offers you good value, proximity to clinical data, and liquidity, which are all pretty attractive. So there's a big focus on the publics right now. In the private space, valuations obviously live a little bit differently. They're probably, frankly, at this point, a little bit closer to reality. I think if you're a generalist, then I think it's always smart to invest in things you understand. So whatever your area of passion where you do have depth, then focus in that area and say, what do I think is an important problem? Do I think this particular technology is going to solve that problem?
I love the computational bio stuff. Anything genetic engineering related. For example, I like to design my own genes using SnapGene, like scaffold them, and then like for the specific like components of the DNA, just there's so many public tools out there that I can use to like piece things together. But really, like the area I love to invest in most is this idea of like simulating new therapeutics and then actually like spinning those out into the real world. So anything from, using tools like SnapGene and then spinning up that therapy with like GenScript, for example, giving them like the essentially the like SnapGene file. Basically anything like computational biology driven, given my background is in software. And the first project that I worked on was a computer vision product that helped people that were blind like identify different objects. And so the area where I personally want to invest in the most is like that intersection of software and biology. But I agree with you on the point of like working with specialists. And one of the things that I think is really important is actually providing that capital to those specialists, a kind of carte blanche, and saying, invest it how you think is best, and giving them economics on that. So that's been a really important part of the fund. We're calling it sort of our like fund of founders. And yeah, someone like you, I would imagine, had they had like a nice balance sheet of like capital to invest, would have been able to spot some pretty exceptional projects.
Well, I'm not an investor, so that's not my primary motivation. Obviously, when you have investors, then clearly your job is to try and give them the return by doing the thing that they've invested in you to do, right? I mean, that's the trade-off, or the agreement you strike with every investor. Really is like, I've been clear about what it is I'm trying to do and why I think it's a good idea and why I think I can turn your capital into 10x. And my job now is to execute on that and try and execute flawlessly. And there'll be bumps in the road, and I'm going to keep going unless I have a reason to believe that it's time to stop.
I guess on that note, I'll let you get back to executing. Andrew, it's been such a pleasure. I really learned a ton, and yeah, keep me posted on what you're doing. It's been so cool.
Now you can find your way around the Marin Headlands. All right.
Yeah, thanks so much for the awesome route.
Good seeing you, man.
Take care. Cheers.